Hey guys, we are going to be starting chapter 14 and we're going to be looking at genetics, and you can't talk about genetics without talking about Mendel. So we're going to get started. Genetics is going to be how we talk about inheritance. So this is the passing of traits from parents to offspring. Humans have known about some type of inheritance for thousands of years, but we didn't actually really understand it until somewhat recently. So genetics is the study of inheritance, or the scientific study of inheritance, and it is relatively new, about 150 years or so old. And again, like a lot of the other stuff that we've covered, it became easier to understand when we developed new tools to understand it. So there are three basic genetic theories that we're going to look at. The first is blending theory. And this theory says that traits are sort of like paints, and so they're mixed evenly from both parents, and so you are essentially a blend of your two parents, of your mom and dad. The second theory is incubation theory, and so this says only one parent controlled the traits of the children, and so evidence for this would be like if all the kids look like mom or all the kids look like dad. Or you've got one kid that looks like mom and another kid that looks like dad, they were in control of that specific child. And then you've got the particulate model. And this one says that parents pass on traits as discrete units that retain their identities in the offspring. So we're going to look at Mendel now and the experiments that he did, and we're going to look at his data and figure out which of these models he agrees with. So Gregor Mendel is called the father of modern genetics because he did so much to start the study of genetics and really do this in a scientific way and to be able to collect data on genetics. He was the first one to do that. He was also a monk, and he published a paper in 1866, but was not really recognized by science until the early 1900s. So why was he so successful? Well, as I said, he used an experimental approach. So he was very specific in the tests that he was running and how he collected his data. He also applied math to the study of this natural phenomenon, and he kept really good records. So he kept track of his data very carefully, and so he was able to go back and really make good comparisons over the different crosses that he had tried. So he was a pea picker, and he used peas as his study organism, which actually worked out really well for a variety of reasons. But it's important because A, they have a really short lifespan, so they can reproduce quickly. They're bisexual. They've got many traits that are known, and so you can look very specifically at several different traits, and you can look at them individually. You could look to see if there's an interaction between traits, so that was really important. You can use them as cross-pollinating or self-pollinating. And of course you can eat the failures. So cross-pollination, what is this? It's when you have two parents, and so it results in hybrid offspring where the offspring may be different than the parents. And so this is really important. Peas naturally self-pollinate, but you can manipulate them in order to cross-pollinate them, which is what Mendel did. So this is a diagram of how he cross-pollinated the plants. So here you have, it says P, or parental generation, so we're going to be calling the first generation the parental generation, or P generation, and then their offspring are called the F1 generation, or first filial generation. And so what you have here are parent 1 and parent 2 is the white flower over here, and you can see that he's removing different parts of the plants. So in this purple flower, he is removing the stamens, which are the male part, and he left the carpel. And then here you can see the white flower has the stamens, which is the male part, and so he's going to cross-pollinate the white flower with the purple flower, and then he's going to look at the data that he gets. And so looking just at flower color right here, if you cross a purple flower with a white flower, what you get is an entire first generation full of purple flowers. So again, self-pollination is when you have one flower that acts as both parents. That is the natural event in peas. So you could see from this last slide that he had to manipulate these flowers in order to get them to cross-pollinate. Typically peas will self-pollinate, and the results are that you get purebred offspring. So where each of the offspring is going to be identical to the parent because it's one flower as both parents, and so those offspring are getting the same genes that were in the parent. So they're going to look exactly alike. So Mendel used 7 different characters, and each of those characters had 2 expressions or 2 traits. So when we use these words, characters and traits, an example would be that character is talking about height, and then the trait would be tall or short. Or a character could be flower color, and the trait would be purple or white, which is the example that we just looked at. This is a breakdown of the different characters. So again, flower color, flower position. Flower color is pretty basic. You just got purple and white. Flower position, axial versus terminal. So terminal would be flower at the top. Axial then is like flowers on the sides, like your arms. Then you've got seed color, green and yellow. You've got seed shape, so round versus wrinkled. The pod shape, the inflated versus the constricted. Pod color would be green versus yellow. And then again, stem length or plant height would be tall versus short. So those are all of the characters that he was looking at with each of the traits for each of the characters. And now what you're looking at here, this is the cross of the F1 generation. Here you're looking at the breakdown of the F2 generation. So the first trait listed in each column, or I'm sorry, in each row is going to be your dominant trait, and then the second one in each row is going to be your recessive trait. The numbers are going to represent the F2 generation. It's a ratio of dominant to recessive, and then finally in this last column, it just sort of simplifying that that ratio, so it's a little bit easier to read, because 705 to 224 doesn't mean much, but if you look at it, it's about 3 to 1. So first, Mendel looked at monohybrid crosses, which we call Mendelian crosses, and these are going to be crosses that work with a single character at a time. So an example would be crossing a tall plant with a short plant, where all you're looking at is plant height. So again, that P generation is the parental generation, or the first 2 individuals that are used in a cross. And so with this example, you'd be taking a tall plant and a short plant, crossing together. They would be your P generation plants. Mendel used reciprocal crosses where the parents alternated for the trait. So if one was tall, the other was short. And then the offspring, or the first generation, you call it F1 or first filial generation. When you cross 2 F1 plants together, then you have the F2 generation, or the second filial generation. And so this can either be done by crossing 2 different F1 plants together, or you could allow an F1 plant to self-pollinate, and then their offspring would also be considered that F2 generation. So in our experiment that we're looking at, again, we have our purple and white flowers, so that is our P generation. When they are crossed, they actually produce all purple flowers. And so our F1 generation, they're called hybrids because we cross 2 true breeding plants. And with 1 purple and 1 white parent, they actually produce all purple plants. And then when you allow or either that 1 plant to self-pollinate, or you cross-pollinate 2 purple plants from that generation, you get the F2 generation, and now you see 3 purple plants or purple flowers to 1 white flower. So we have that 3 to 1 ratio that I mentioned a second ago. So his results showed that in all crosses, the F1 generation showed only 1 of the traits, regardless of which was male or female. And then the other trait, the one that disappeared in that F1 generation, then reappeared in the F2 generation at about 25%. So it ended up with a 3 to 1 ratio in that F2 generation. So Mendel's hypothesis had several parts. First, he said genes can have alternate versions, which he called alleles. So each offspring inherits 2 alleles. You get 1 from each parent. So what that means is that F1 generation was getting 1 purple allele from the 1 parent and the one white allele from the other parent. So that offspring would have 2 alleles. He then said that if the 2 alleles differ, then the dominant allele is expressed, and then the recessive allele remains hidden unless the dominant allele is absent. So in our example, we crossed a white flower with a purple flower. We had an F1 generation of all purple flowers. So that means purple would be dominant, white would be recessive. The white disappeared from that generation, but then when you cross 2 individuals from that F1 generation together, then about 75% of the offspring will be purple, and then about 25 will be white. So that recessive allele sort of reappears then in that next generation. So just a quick comment, when you guys are talking about alleles being dominant versus recessive, make sure you don't refer to them as like strong or weak to describe dominant versus recessive. You should be using the terms dominant and recessive. And then finally, the two alleles for each trait separate during gamete formation, and now we call this Mendel's law of segregation. So the law of segregation shows, if we look at again, that P generation, the purple flower is going to have two capital P alleles. P is for purple, and so we show the dominant allele with the capital letter, and then instead of using a W for white, we just show a lowercase p for the white flowers because that's recessive. So the purple flowers contain the two capital P or dominant alleles. The white flowers have the two recessive alleles. When the gametes are formed, you're only going to have one allele per gamete. When these two fuse together, you now have offspring that have one dominant, one recessive allele, and so they're going to show the dominant form, which in this case is purple. Now, when these alleles are made, half of them will be, half of the alleles will contain the dominant, the other half will contain the recessive, and so now when you cross to these two members of the F1 generation, what you end up seeing then is 3 purples to 1 white, where when you look at the genotype, you have 1 homozygous dominant, 2 heterozygous, and then 1 homozygous recessive. So I just threw a bunch of vocab at you that you haven't heard in a few years, and we're going to get to that in a second. So Mendel's experiments showed that the particulate model fit the results the best. And some vocabulary that I just used that I want to make sure we review quickly. Phenotype is the actual physical appearance of the organism, so just remember pheno and physical, so it's what that organism looks like. Genotype then is the genetic makeup of the organism, and it's usually shown in some type of code, and we usually use alleles to show that. So if you had 2 capital P's, the genotype would be 2 dominant alleles. If you had 2 lowercase, it would be 2 recessive alleles, and so we tend to show the genotype by writing out the letters for the alleles. All right. So again, we've got our 3 dominants to our 1 recessive, and we also have these words here that I already said. So homozygous dominant is up here because you've got the 2 capital P's. The homozygous recessive is down here because you have the 2 lowercase, and then you've got 2 heterozygous in the middle, and we call them heterozygous because they have 1 dominant and 1 recessive allele each. So homozygous is when 2 alleles are the same. So again, you can have homozygous dominant, which is capital T, in this case for tall, or you can have homozygous recessive, which would be lower t, in this case meaning short. And then heterozygous is going to be when the 2 alleles are different. And so here you have 1 dominant T and 1 recessive T. Individuals that are homozygous dominant are going to be dominant. Individuals that are homozygous recessive will show the recessive form. And then individuals that are heterozygous, because they have that dominant allele, they will show the dominant phenotype. So in the case of the T's, that means that the individual that is heterozygous would be tall. Alright, so that is the end of this first intro lecture into genetics. And so your lecture question for today is, please explain in your own words why the particulate model was the most accurate model based on Mendel's data. Alright guys, thank you very much, and please make sure that you respond to this question via the class website. Thanks.